So thank you for the invitation to present this new center, the Quebec Genomic Data Center, CQDG in French, which my team is developing and maintaining, running at the research center of the St. Justine University Hospital here in Montreal. So the CQDG is what we used to call a genomic data commons. In one sentence, genomic data commons can be defined as a secure platforms for hosting and sharing harmonized data from many genome research and clinical studies. So I've been working on a few of them over the last few years. You already mentioned them almost. So ICGC and the NCI GDC on cancer, the NIH Gabriela Miller Kids First Data Resource Center on pediatric cancer and birth defect, the NIH Include project on Down syndrome, and now on the upcoming PCGL, which is the Pan-Canadian Human Genome Library, that should be, for which we expect starting its operation somewhere in 2025. So all these commons, except the PCGL, of course, are currently making accessible 1,000 of genomes to the research community, and this is really the primary goal of a data common. So the CQDG was launched 2 months ago only, in May, and its mission is really to consolidate in the same data infrastructure a critical mass of genomic data to enable research in precision medicine and AI applied to genomic in Quebec. So we are a generalist platform, meaning that we're not tied to a specific disease or a specific study design. And although our primary focus is to support the Quebec studies, we are more than welcome to accept and host data from elsewhere in the world, valuable data sets from elsewhere in the world. We succeeded to secure some funding, 10.5 million Canadian dollars in total. We received money from Genome Quebec and the Fonds de Recherche de Québec en Santé to support our salaries. And the hardware comes from the funding of the hardware comes from the Canada Foundation for Innovation, more specifically through the Secure Data for Health project, which is a project coded by Guillaume Bourque and myself. And one of the main aims of this project is to build an academic cloud compute infrastructure, specifically designed and governed to store or host sensitive health data. So we are in our second phase of hardware acquisition, and early next year our cluster, OpenStack and Ceph cluster, should include more than 10,000 CPU, 20 petabytes of storage, and so forth. This hardware is stored and managed by Calcul Québec, which is an organization in Quebec to support the research needs of the compute need of the research project in Quebec. So let's talk about our first data release. We have currently data on 3,300 participants from 5 different studies, which include Cartagena, which is an important population longitudinal cohort of 40,000 participants in Quebec. We have 2,100 whole genome from them. And we have also other studies on rare diseases and neurodevelopmental disorders. This may seem small, but believe me, the most difficult release in a project like this is the first one. So big or not, it doesn't matter. And but anyway, so we aim to frequently release new data sets. We will have new genome and exome from the current contributing studies. For example, Cartagena is looking for sequencing its entire cohort. We will recruit new contributing studies, of course. We are a partner of the new Quebec Rare Disease Network, for which we will host and store the data. And as an example of a data set that is not from Quebec, I am in discussion with Melanie, she's not here, but from YCR, to host the somatic data of 20,000 donors from the ICGC phase 1. But the big thing that will make the CQDG different and very interesting, it's really its integration with the Quebec public healthcare system. So in Quebec, we have adopted a model similar to those in UK and France for clinical genomic, And this model is illustrated here in this blue box. So patient molecular diagnosis test is handled by a network of 9 laboratories in Quebec, across Quebec. But the whole genome and whole exome sequencing is done centrally at a center called the Quebec Genomic Center, which is located at St. Justin, my institute. And so this center is not only responsible for sequencing, but is also responsible for bioinformatics. So we are responsible for archiving the sequencing raw data, calling variants, annotating them, and storing them in a queryable database, a variant database that can be used through a web application by the lab geneticists to interpret their variants and to produce their report. So the nice thing here is that this data will be copied to the CQDG research environment for the consented patient. So that's very, so when this will be up to speed, we will have I wanted to say, so once it's important to the CQDG, well, this data will get harmonized and with the other CQDG data set and made available to the research community. So when this will be up to speed, we will have 1,000 very clinical grade exome, genome, and new genome annually to the CQDG, which is quite amazing. And this genome will come from patients with a variety of medical conditions. So one slide on the governance. So our governance framework was written by the McGill Center of Genomic and Policy at McGill, which is a leading group in the field of genome governance. An important point to keep in mind is the studies continue to control who have access to their data set, even if this data set is hosted on the CQDG platform. So the CQDG just act as a broker and execute simply the decision made by the studies data access committee. But we request that the CQDG, the data set, the study data set are described, the user, what do you say that? The data set permission, use permission, and rules of constraint are described by the ontology, the GeoFo ontology, data use ontology. So which are listed in this yellow box. So this is really important actually because it will help the researcher to quickly determine if a data set is compatible with their use cases. So for example, a big pharma will want to access a data set. Well, if this data set is tagged with the term at the bottom, non-for-profit organization use only, well, they won't make any data access requests for this data set. So that's important. So let me quickly give you an overview of how it works, the CQDG works. A very broad overview. So on one end, you have the clinical data that is submitted to the data coordinating center. Just between us, the data coordinator currently is one person. So, so it's not a big center, but it does the job. So, so the clinical data gets validated, converted into a specific format, and imported into what we call our knowledge base. The DCC is also responsible for user support and documentation. On the other end, the molecular data is processed by our bioinformatics teams. We want to process all the molecular data as much as possible in the same way, uniform way, especially for the variant coding step. And once this is processed, it produces new files that are imported on our CQDG platform in our object store and made available to authorized researchers. Also, the variants are extracted from the VCFs and stored in a variant store also on our CQDG platform. This platform has also other components like the data portal, variant workbench, secure compute cloud, and bioinformatics. Now, for the rest of the presentation, I will give you details on each of these components. So let me start by the clinical data harmonization. So this is achieved through what we call the CQDG dictionary. So the CQDG dictionary specifies the clinical data elements and biospecimen data elements that are submitted to the CQDG or variables. So it includes, because we are a generalist platform, so we need to support different variable relevant to different research domains, such as cancer, rare disease, population study, and so forth. So the submitted data must comply with the CQDG dictionary. So it's mandatory. So we enforce the use of ontology in our dictionary, so such a human phenotype ontology, MONDO, for diagnosis. And the variables are grouped in 12 tables in our dictionary that are listed here in this yellow box. So you have dataset, exposure, follow-up, phenotypes, family. So let me show one of this table, the phenotypes one. This visualization is available on our online documentation center. It's provided by a software called Lectern, which is developed by OICR. It's open source, and we're using it. So you can see, for example, this table has 6 variables. You can see the phenotype HPO code. No, there's no pointer. So you can see the phenotype HPO code. This is where we harmonize our HPO. So yeah. Yeah, it will take too much time. Okay. Yeah, so you see the phenotype HPO code is required. So you have the column whether indicating whether a field is mandatory or not. And the HPO code is mandatory. So every phenotype that will be submitted by the studies will have to correspond to one or more HPO code. This is how we achieve harmonization for the phenotypes. And we do the same for the diagnosis with the MONDO ontology. So you see also in this table that you have the validation rows. These are regular expression, but you can have also more complex algorithm. So not only the Lectern provides this visualization, but also it acts as a validation system. So it will apply these rows to the data that has been submitted by the submitter and will return errors if the validation failed. So not only we in addition to using ontologies, with the CQDG, our platform, we aim to be interoperable with other data platforms worldwide. So we have adopted many standards for that to achieve this goal, including one which is called FHIR. So FHIR stands for Fast Healthcare Interoperability Resource. So it's a HL7 standard for healthcare data exchange. So not only it specifies the way the data should be formatted, but it specifies also the way you can access or query this data. So it's an API, basically. So it's adopted by many big players in the healthcare industry, like Epic and Apple Health. And it's part of the NIH Cloud Platform Interoperability, NCPI, which we are part of this effort. And the aim of this effort is to create a federated genomic data ecosystem. So how does it work? So once the data is validated with Lectern, it's converted and imported into our FHIR server. And then we can this FHIR can be interrogated using the standard FHIR API. So the molecular data harmonization, well, as I said, we aim to, as much as we can, reprocess the data, the molecular data we receive, especially the variant calls, in a uniform way across all the platform, all the studies. So our bioinformatics platform is built upon Nextflow and use, for example, some pipeline from the NF Core library. One thing that is very nice with Nextflow is that you can it's a platform agnostic. So it means that you can, the exact same workflow can run on HPC, a cloud, or a Kubernetes cluster without any change in the code. And this is very, very nice for us, because we actually do have access to an HPC and a Kubernetes cluster. So we can run our flow faster on more sample since we have these two resources available for us. So it's very useful for us. And also the fact that it's platform university, maybe it encourages usability by other bioinformaticians. So how it works, so we receive the files from the submitter. We do some data integrity check, also using Nextflow. We run the pipelines, the variant calls, sequence alignment, and so forth. We do have a step for variant annotation, and all this is imported for the metadata in our knowledge base in FHIR. With the files are stored on our object store, and the variant calls in our variant store. So current pipeline now we have DNA variant calling annotation, de novo variant identification for family analysis, RNA-seq short and long read, and RNA splicing analysis. So variant annotation, well, we try to add as much as we can annotation. Actually, it's an ongoing task. Every month we have new annotation added into our pipeline. This is simply a list of classical annotation, I think. So predictive function annotation, like the usual scores, CIF, SDMM, CAD. We do have Splice AI for the alternative splicing. We have clinical significance. We import ClinVar, VEP, gene phenotype association panel, HPO for that. And also, we also import all the public frequencies produced by Nomad, TopMed. And we have also our own annotation. So we calculate the allele frequencies within the CQTG cohort and made this available. The variant store, all this variant annotation, the variant are stored, as I said, in our variant store, which is obviously a NoSQL database given the size of this data. So the data is, so this database, this store, not only has the variant data, but is also enriched by the partisan clinical data that we have in our knowledge base. And also it contains all the public external database that we use for annotation. So we have Nomad, ClinVar, name it, we have it, hopefully. So all this into a same database, queryable database that is run using or built using technologies like Parquet. These files are Parquet files are managed using Delta table, and then all this is running on a Spark cluster for distributed data processing. So and at the end, we indexed all this content using Elasticsearch, which provide very responsive query for the front end. Currently, our database has 90 million unique germline variant. And since this variant can occur in multiple patients or participants, this is what we call non-occurrence. We currently have 13.5 billion germline variant occurrence in our database. We know that this can scale up because in Kids First project, we're using exactly the same project, the same framework. And we do have 100 billion occurrence in our database for 300 million variants. So we're not worried about this. To access this data, we have a variant workbench. Currently, it's limited only for internal use. We haven't exposed it to the researcher for some technical reason that we will solve later this year. So with this notebook environment, it's an Apache Zeppelin notebook environment, where you can do everything you want using the language you like, SQL, Python, Scala, Markdown. And we do have a pre-installed libraries such as AL, Panda, Navi, and so forth. So this is ready for our QC now. And then eventually we will open it for the researchers. Now for downloading data, we have developed a tool called Failload. So the usual workflow is the following. So as a researcher, build a virtual cohort, find the file of participant, find the files that are of this participant they are interested in. They request data access. They make a data access request. Once it's approved, they can go to the portal, download a file manifest, and use it to do a bulk download using this tool. So this tool is connected to our OAuth system, Keycloak, and we're using multi-factor authorization, of course, for downloading the sensitive data. But one thing that is very nice with Fairload is its compliance with the GA4GH Data Repository Service, or DRS, which make the CQDG interoperable with any other platform, like Cavatica, and I'll show you in a minute what I mean exactly by that. Let's talk about the public-facing component of our platform. So we have three parts. We have a website, which is simply a static web page. We have the data portal and a online documentation center. So today, of course, I will talk about only the data portal. So the data portal current version has many features. So we have a study repository. We have a virtual code builder. We have a germline a search tool for our germline variant database. We have an entity page. So each entity that we have, study, participant, files, variant, has its own entity page. We have user custom dashboard and so on. So I'll go through some screenshot now. So this is the study repository, which is a list of the 5 studies we have currently. Of course, when we have more study, it would be more interesting. But this table gives general information on each study. For example, you have the domain, rare disease, general health, and so forth. We have the number of participants per study, the number of family per study, and we have the kind of data the study produce. For example, now we have only studies that are focusing more on the whole genome sequencing or whole exome sequencing. So about only the genomic data, but we expect study with transcriptomic or imaging eventually. This list can be filtered and searched using the facet as usual on the left. You can notably search by access limitation and access requirement that are also encoded using the duo code. When you click on the link for each study, you get the study entity page, which contains a lot of information divided in different sections. The first section is the summary section, where you have description of the study. You have some keywords, the domain, the type of population, it's adult or pediatric or both. You have a data access section. Again, all this information is encoded with the duo for access limitation, requirement, and authority. Also, we have a data file section, which summarizes which gives the number of files the study have broken down by different data types. So align read, germline structure variant, and so forth, and broken down by experimental strategy like whole genome, whole exome. And we have also the concept of data set. So a data set is a subset of the study data that the study really wants to highlight for the researcher. It can be, for example, a data set from a publication. And so the study would like to highlight this data set and make it accessible very quickly. So the researcher can go directly to this page and request access to this specific data set only in a quickly manner. This is our data exploration or cohort builder. So that's a complex tool. I don't have time to explain all of the detail of it. But briefly, this tool is really meant for researcher or user to build virtual cohort of participants from multiple studies based on search criteria. So the nice thing with this tool is that it searches simultaneously the participant, their biospecimen, and their files. So on the left, you have the facets, as usual. You are divided in 3 categories, facet relevant to participant data, like clinical, relevant to biospecimen, and relevant to data file properties, like the data types. And this left panel can be used to add terms on the query builder here on the top. So you can formulate very complex queries. You can have multiple, you can add as many queries as you want. You can combine them with logical operator or an hand, for example. Once you're happy with your filter, you can save it. You can name it, and you can even share your filter with other users of the portal. This at the bottom is what we call the summary view, which is a graphical representation of the result of your query. These graphs represent different facets and are clickable, the dynamic, and so forth. So, So now I'm going to explain you how we leverage our effort in on clinical data harmonization. Remember, all the phenotypes and diagnosis Mondo are encoded using Mondo and HPO. You see, so when you open the facet here on the left, you have phenotype HPO diagnosis. When you open it, actually, you obtain a built-in ontology browser that we built, actually. This is a HPO tree that you can obtain with participant count. So you see that you can navigate through the hierarchy of the ontology. For example, this node, abnormality of the head, is a parent of abnormality of the face, which is also a parent of the abnormality of the mouth. Okay, and for each term, you get two participants that for which with a phenotype that match exactly this term, and the number of participants that match this term, but also all the children term of the ontology. So when you do a query, we actually find all the participants having phenotype with the term exact and the children term. For example, children term, sorry. So for example, in this result table, you have a participant with the narrow mouth and depressed nasal ridge phenotype. Well, we successfully found this participant simply by looking for a phenotype abnormality of the face. Whoops, sorry. So you see, so when you type it, when you search for a term, you find also the participant with the terms for the children of this coded term. I'm not sure I'm clear, but so yeah. Okay, thank you, David. But this is really important because when we receive data from different studies, they don't have the same level of precision or granularity in their phenotype. So it's very important to be able to navigate with this different level of precision, and this is the tool to do that. We have the same tool for the diagnosis as well. So the second view, in addition to the summary view that I presented earlier, we have the participant view. So this is a table of participants with their phenotypes, diagnosis. This table can be customized with this little wheel here. You can add more columns and remove others if you want. You can, so for example, and remember all this. The query can be a mix of different properties. So for example, this query we have a clinical diagnosis, nervous system disorder. We have some clinical demographic data like the criteria, some age of recruitment, sex, ethnicity. And then we have the data type, which is a file property within all the same query. So we request participant who has germline structural variant data. This is all in the same query, and this is what you find. You find these participants, but you will find when we open the data file view, the corresponding files for these participants. So you can, as I said, configure this table. You can select rows and download the clinical data associated with this participant. And if you click on the participant, you get the participant at its page that will give you more detail on each participant, on the specific participant. So this is the biospecimen view, which is similar to the participant one, which is a list of biospecimen criteria that correspond to this query. And again, it's a configurable table. In another project, actually, we add a button. So the same way you can request for access to data, here we can request access to the biospecimen, not here, but in the other project. We have a tool to request access to biospecimen, and these requests are sent to the biobanks represented in this list of, so it can be multiple biobank in the same list of participant, of sample. Again, this sample can be filtered by type, by tissue type, age at the biospecimen collection, the sample type, and so on. So the last view is our data file repository, which is the most important one. So again, you have a table of the list of files that correspond to your query. You can filter it down more by data types, data category, data set, strategy, experimental strategy, format, BCF, and so forth. And then what do you do with this table? Well, you can select specific rows if you want. And for this selection, you can either save it as a file set. Actually, for participant, biospecimen, you can save your list as a participant or biospecimen set, but you can do the same for the files. You can download a manifest. So this manifest will be used by Fairload to download these lists of files, bulk download. And you can request access. Before, actually, you can request access. So the request access button will create an archive for the researcher that contains all the information necessary that can be shipped to the different studies involved in this list of files and for their data access requests. So it's a tool for helping the researcher to formulate their data access requests to multiple studies, the multiple studies that involve in this list. And finally, you can push, click on the analyze in Cavatica. I'll show you in a minute what it means to analyze this selection of file into the Cavatica environment. So here, quickly, I don't have much time. So quickly, this is our variant database. As I said, we have 19 million unique variants now. This shows only aggregated data. You don't have the occurrences because it's control access data. So we have an aggregated view of our variant store here. I won't go into details. So we have 34 search criteria now available that are grouped, divided in 5 categories on the left, the blue panel. Here, for example, this variable, this table, this query is so it's all the variant pathogenic in ClinVar with that are rare, like 5% allele frequency, less than 5% allele frequency in Nomad. And then you have the table. We show the most dangerous consequences in this table, but of course there are consequences for each transcript, and this can be obtained or viewed in the variant entity page where all the details is displayed here, including all the transcript consequences and so forth. We do have our own annotation, so we calculate the frequency, the CQDG frequency per Brogan-Dunbar study. Finally, we have a little page for user community. So when you register to the portal, you can add your affiliation, role, and research interest. And this is a really simple interface to try to create a community of researcher that are using the portal. Now, I'm almost done. The CQGG in the cloud with Cavatica. So Cavatica is a platform a bit like Galaxy, so similar, but it's maybe not similar, but they have the same goal. So it's provide a compute environment with a lot of tools for researchers to work with. So it has a different kind of tools. So you have the RStudio Jupyter for prototyping and effective analysis. You do have also hundreds of different workflows already pre-made, ready for production, and you can just upload your data and run those workflows automatically using this framework or this website or this infrastructure, let's say. And you have also a different kind of web app application, web app tools like Shiny apps that you can build directly from this platform. So the idea, since we are DRS compliant, which is wonderful, so we can, so you don't want people or researchers to download under the BAM files on their laptops, on their server, would take forever and so on. So the idea is that we interoperate with Cavatica so that the researcher can access the file they have access to on the CQDG directly from Cavatica without any transfer. It's a virtual file system that is mounted on the Cavatica side, and then the researcher can see all its file that is authorized to see in the CQDG. So this is what we can do with DRS compliant platforms. And I was wondering when I was listening to Galaxy Cloud if Galaxy was DRS compliant. Is it? No? Okay. Too bad, because we could have connected Galaxy to the CQDG, but maybe in a few months. So basically, when you are in the file repository, You select the files, as I said, and then you click on the push Cavatica, and then you have this window that will pop up, and then you will see the kind of files, the number of files broken up by studies that you have access to, and then you click on the copy file, and when you go to your Cavatica workspace, you will see the CQDG files. The same way you will see other files that you have access in other DRS compliant platforms, such as KidsFirst, Include, Handville, and so on. So you can upload your data and access different data sets from multiple platforms if you have access to. So the next objective for the coming months, let's say, so we will work closely with the research committee to recruit more studies and add more data set to the CQDG. We will develop 2 new web apps, one for the data submission, to help the studies to submit their data. And we need to build a participant portal in collaboration with the Quebec Rare Disease Network. We will extend our variant database to support somatic variant. And then, as I said, we will probably give access to the variant workbench to the authorized researcher. And finally, and not the least, we want to expose individual data to authorized researcher within the portal, which means that we want from the variant database, variant interface, for example, we want to be able to see the occurrences. So if you have access to one genome, you want to see the 4 million variants that the genome has, and start working with it, make some queries, filter, and so on. Do some research using the CQDG portal, and not using simply as a catalog. So this project will be done in collaboration with the Children's Hospital of Philadelphia and Sick Kids. In conclusion, so genomic data commons like the CQDG play an essential role in precision medicines by making harmonized genomic data set discoverable and available to researchers in the secure computer environment. The CQDG really wants to harmonize its data, so it uses data ontologies and implements standards such as FHIR and DIRS to make sure to ensure interoperability with other platforms like Cavatica, and there will be other coming soon, not soon, but this year. And finally, the CQDG is the research arm in the Quebec strategy on clinical genomic by making the genomic data, clinical genomic data, and exome and public, not public, but available from Quebec patient, available for research. Finally, I would like to thank my team. This slide, actually, I realized that it's misleading. It's not all the CQDG team. This is my entire team. And a few people, well, almost everybody has touched the CQDG platform more or less. And I want a special thank to Jeremy Costanza, which is our tech lead and software architect. And of course, I want to thank our funding agency. Thank you.
We have a couple of minutes for maybe 1 or 2 questions. So if anybody has one, please maybe just come here, or there's a microphone on the other side.
Merci, Vincent. The alignments and the mapping of variants and so forth, will that be using what Guillaume was talking about this morning, the pangenome reference?
Not
in the near future?
No.
Like not for years?
I don't know, actually. Okay. I need to talk with Guillaume, actually.
You mentioned the cloud environment in AWS in particular.
Okay, sorry.
Okay, thank you. You mentioned the cloud environment to provide a cloud environment for researchers to use. Is that researchers would have to bring their own funding to pay for the cost, or?
You mean Cavatica?
Yeah, Cavatica.
Yeah. So the nice thing is actually it's outside of the CQDG. Yeah. So to use Cavatica, you need to there are some fees actually. Yeah. So
okay. And then do you allow the researchers bringing their own tools into the cloud environment?
Yeah, they can do that on Cavatica as well.
Okay, so. Thank you.
Great talk, Vincent. It's nice to hear this project come to fruition. You mentioned about bringing in ICGC data, but if I recall correctly, ICGC data doesn't have clinical aspects, and so is that a barrier for?
No. Yeah.
Okay. Because it
We'll
would
find
just
out.
be the genomic data, right?
Huh?
It would
No,
just,
but we still have the cancer type can be mapped to Edmondo diagnosis.
okay.
This kind of, but I'm very aware that
Okay.
it's
Yeah.
quite limited.
Yeah.
So,
Sure.
But yeah,
Okay.
well, we will map. We'll find a way.
Thank you for the great talk. So you mentioned the genomic data sharing platform accumulates samples, right? So you're not recalling the whole cohort. You are processing sample by sample with the pipeline with Nextflow and ingesting variants into the platform. So how do you consolidate variants from different samples? How do you make sure that one sample has coverage in this locus?
Yeah, so we do some QC to make sure the coverage is there for sure. And then we do a joint genotyping as well. And also we are currently implementing HALE, and we will maintain a variant matrix for the entire database. And this is how we will, this is not done yet. But
Thank you. And are you planning to switch to Dragon at all, or will you stay
Dragon,
with?
you said?
Will you switch to? Are you planning to switch to Dragon variant calling at some point, or will you stay with JDK?
Okay. So most of the data now that we receive are from the beginning are called with Dragon, and we haven't touched that. So here's a discussion. Actually, I said we will try to harmonize as much as we can, but if 90% of the data we receive are using Dragon, I think there's no need to harmonize. We will harmonize the 10%. Maybe we'll acquire a Dragon server. I don't know yet, but yeah.
Okay,
but
thank you.
Yeah. Ideally, I would like to reprocess everything and not using Dragon. And we're building the infrastructure for that aim, actually.
Thank you.